Namaskar. Today we'll talk about protein synthesis or translation. Protein synthesis or translation is the final step of central dogma. That is flow of information from DNA to protein via RNA. We have talked about this central dogma many times. So this last step, the synthesis of polypeptide or protein by reading the genetic code in the mRNA is called protein synthesis. It is the process of gene expression actually. So let's first see how the protein synthesis differs in prokaryotes and eukaryotes. Just an outlook of that, where does it occur? Grossly, how does it differ between these two types of organisms? In case of prokaryotes, there is no nucleus and therefore the chromosome is present in the cytoplasm. It is floating in the cytoplasm along with the ribozymes, the methionyl for protein synthesis. So that's why when transcription is going on, the ribosome gets access to bind with the transcripts and initiate the process of translation. So in case of prokaryotes, transcription and translation, these two processes are coupled. This is also because the genes in the prokaryote system are continuous. They are not separated into coding and non-coding regions. So whatever present, it all is coding region. In fact, a number of genes are consecutively present to form operons because the space in case of prokaryote system chromosome, single chromosome containing so many genes is very, very limited. For example, in E. coli, it contains 4,000 genes in a single chromosome. So the genes has to be present very closely on the chromosome. And more often they're present in the form of operons. In case of eukaryotic system, there is a nuclear membrane and the genes are present, the DNA is present within the chromosomes which resides in the nucleus. The transcription occurs within the nucleus where the RNA is formed from DNA by transcription. That RNA in fact is an unprocessed RNA, that is a premature RNA, also called heterogeneous nuclear RNA. So this RNA contains non-coding regions also called introns. The coding regions are called exons. So non-coding regions have to be removed. And then the RNA, the mature RNA, that will be transported outside the nucleus and the protein synthesis will occur in the cytoplasm then. So in case of eukaryotic system, transcription and translation, these two processes are spatially and temporally separated because of the nature of the genes, because they are discontinuous, split gene. So now let's move to the important protein synthesis mechanism in case of prokaryotic system, particularly in Escherichia coli. This is a typical organism in which protein synthesis has been studied in detail. So what is required for protein synthesis? The first thing that is required is the ribozymes, which are the methionyls for protein synthesis. Next thing will be the information carrying molecule, those are the mRNAs that contains genetic codes. And then the tRNAs that can read the genetic codes are required and they bring the amino acids, corresponding amino acids. There are 32 types of tRNAs in the cytoplasm and for 20 different amino acids. The number is more because of the codon degeneracy, and in fact there are different types of initiator and elongation phase tRNAs. The initiator phase tRNA is called tRNAi and the elongation phase tRNAs are called tRNAm. In case of both prokaryotes and eukaryotes, the initiator tRNA, initiator amino acid is the methionine. So initiator tRNA is met-tRNA. Methionine binding, methionine acylating tRNA. The next step, next thing that is required, of course, are the amino acids, the ingredients, the initiation factors, the elongation factors, the release factors for initiation, elongation, and termination phases of protein synthesis. A number of GTPs are required in many steps in protein synthesis. We will see them soon. And ATP is required in the first step when tRNA are being attached to their corresponding amino acids. The process is called charging. and a number of enzymes like the amino acid tRNA synthetase, formal transferase, peptidyl transferase. Peptidyl transferase. This is an enzyme that resides in the 23S rRNA of the large subunit of ribosome. So basically, this is a ribozyme. The translocase, which moves the ribosome on the in a 5' to 3' direction towards the next codon. This enzyme is present within the elongation factor G, in fact. So the first step of protein synthesis before initiation is the preparation for the process. And in the preparation, what happens is the amino acids that are present in the cytoplasm, they has to be tagged with the corresponding tRNAs, because tRNAs are not bound to the amino acids at the first instance. So each of the amino acids, 20 different amino acids, those are proteinaceous amino acids, they have to bind with the corresponding tRNAs. This process is called amino acylation of tRNA, and this occurs by a process called charging of tRNA. What is this charging? The charging is energization. It is a process of adenylation, basically. An adenyl molecule, the AMP residue from an ATP, is added first to an amino acid at its carboxyl end. The enzyme required is amino acyl tRNA synthetase. And the ATP undergoes a pyrophosphate cleavage, and later on this pyrophosphate is also hydrolyzed to inorganic phosphates. So basically this is equivalent to 2 ATPs that we require in this step. And next step, what happens is this adenylated amino acid is transferred to the corresponding tRNA at its 3' end. The 3' end of the tRNA contains a CCA residue. The terminus residue, that is A, the ribose subunit of that, at the 3' position, this amino acid is being tagged like this. There are in fact two processes, class 1 and class 2. There are two types of amino acid tRNA synthetase. In case of class 1, the amino acid is first bound to the 2' position, and then it is shifted to the 3' position. In class 2, it is directly shifted to the 3' position. So finally what happens is that the tRNA molecules for each of the amino acids that are required for protein synthesis have to undergo this process of these reactions, and they have to tag with their corresponding amino acids. This process is called amino acylation of tRNAs. The next thing what will happen is the initiator tRNA, that is tRNA, has to be converted into a formylated form. The methionine has to be formylated by a reaction called transformylation. The enzyme required will be transformylase. The formyl group will be transferred from formyl tetrahydrofolate and to the amino terminus of the methionine residue that has been acylated to its tRNA. So it will finally form, this is the formyl group tagged to the amino terminus end of the methionine, and it will form F-met tRNA, F-met. So this is the initiator tRNA, amino acylated tRNA rather. So like all processes of polymerization, like replication, transcription, protein synthesis is also a polymerization process. So definitely there will be an initiation phase, there will be an elongation phase, and termination phase. In the initiation phase, what is required? The initiation phase, basically, definitely there will be an mRNA that contains the information, genetic codes that has to be there, and the ribosomes have to be associated with this mRNA. This is the first step of initiation. How does it occur? It occurs in a stepwise manner. First, the 30th ribosome subunit, the small subunit, it binds with the 5' end of the mRNA, and it moves in the 5' to 3' direction to find a particular sequence called Shine-Dalgarno sequence. The Shine-Dalgarno sequence is a recognition site. Just after that, there is an initiator codon AUG. So the AUG, particular AUG, which is associated with the Schindler-Garner sequence, is the initiator AUG. Otherwise, any other AUGs that are present in the mRNA are not an initiator AUG. So the 30th ribosome will find this Schindler-Garner sequence with the help of its 16S rRNA that is present within it. The 16S rRNA contains the complementary sequence for this Schindler-Garner sequence, and as soon as it finds it, it hybridizes with that sequence and it pauses there. Along with the 30th ribosome subunit, initiation factor 1 and 3, they also come in associate with the 30th ribosome. The next step is the binding of the F-met tRNA F-met to the peptidyl site of the 30th ribosome. There are three sites. This site, the middle site, is called peptidyl site. The empty site on both sides are there. On the downstream, there is an A site, amino site or acceptor site, and the second tRNA will come and will bind, and then there will be a peptide bond formation between the two amino acids. And this is the exit site when the ribosome will move one codon further on the 3' end side, on the downstream side. Then what will happen? This tRNA will become empty and it will come to this exit site and it has to be removed thereafter. So the next step in initiation is the binding of the 50th ribosome subunit. So that the 70th ribosome, the complete ribosome formation is completed. So 50th ribosomes come with and bind with this complex. In fact, when F-met tRNA F-met comes, it comes with the GTP and initiation factor 2 also. So when 50th ribosome binds, the GTP is hydrolyzed to GTP and PI, and the initiation factors are released one after another. So this completes the initiation phase, and this is called the initiation complex or the ternary complex because it contains three things: the mRNA, ribosome, and the tRNA. So now let's move to the elongation phase. In elongation phase, we were here in this initiation phase, we were at this position. So the first tRNA, the F-met tRNA F-met has come and associated with the peptidyl site. Then the next tRNA has to come and bind to the acceptor site or A site. That happens with the help of elongation factor TU, which comes with GTP, binding with GTP, and it puts the tRNA, second tRNA, amino acid tRNA, on the acceptor site, and at the same time it gets hydrolyzed. The GTP gets hydrolyzed to GDP, and the elongation factor is released. So elongation factor is further recycled. The GTP is replaced by another elongation factor called EFTS. So within this cycle, elongation cycle, an EFTU-EFTS complex is formed in the middle. This step is jumped here, and after that what will happen? Again GTP will replace the EFTS and will take its place, and it will form EFTU-GTP, which will bring the third amino acid in the elongation phase. Like this, third, fourth, fifth, sixth, until the elongation process, until the termination site arrives, this process will go on with taking one after another amino acid. What happens between these two amino acids or amino acid tRNAs? The two amino acids form a peptide bond between them. By a reaction, we will see in the next slide how it occurs. The main thing is this, that the first tRNA, it transfers its amino acid to the second amino acid, second tRNA, amino acid tRNA, via a nucleophilic attack, and a covalent bond between them is formed, that is called peptide bond. This reaction is carried over by an enzyme called peptidyl transferase, and that enzyme, as we saw in our second slide or third slide probably, that resides in the 23S rRNA of the large subunit of ribosome. So the two amino acids have been, the dipeptide has been formed, and now the ribosome will move to the next codon. So when the peptide bond has been formed between these two amino acids, and the ribosome has to shift to the next codon on the right hand side, on the downstream, towards the 3' end. And this process is called translocation, and the enzyme, this is an enzymatic activity, and this activity resides in the elongation factor G. One molecule of GTP is hydrolyzed at this step, and the ribosome moves one step further to the next genetic code. When it happens, then what happens is that these two tRNAs, the MT1 and this dipeptide tRNA, this tRNA move from A to P site, A and P site to P and E site. You can see they move one step, one genetic code on this side. When they're moving, this one will come to this position and this will come to the second position. So now the empty tRNA is on the exit side and it will be released from this place. And the A side is again empty, and the third amino acid tRNA will come and associate here. And again, a nucleophilic attack will occur between the third amino acid and this dipeptide, and these three amino acids will be covalently tagged. And again, the ribosome will move to one codon, the fourth codon on the downstream, and this tRNA will become empty then, and it will come to the exit site and will be released. This process is called elongation, and it will continue till the stop codons, the three stop codons, termination codons UAA, UAG, and UGA, they come in the way. However, an ORF, the open reading frame, is the minimum size of the open reading frame is supposed to be 150 nucleotide residues, and so that the 50 amino acid long polypeptide is being formed. So it has to be seen that if it is in true sense the protein is functional, then at least 50 amino acids will be tagged, and the termination codon will be present after that only. So this process is called elongation, and now let's see how the process termination occurs. Before that, this peptidyl transferase reaction, a little bit about that in details. So peptidyl transferase reaction is the reaction between the amino acid and between the peptide and the amino acid, the new amino acid, you can say. So this is here the two structure that has been shown is the first formylation nucleon, the first amino acid, and the second amino acid that has been brought by the second amino acid tRNA. But this process will occur again and again as many amino acids will be tagged with the growing polypeptide. So what happens? This amino group, the nitrogen atom of the amino group contains a lone pair of electrons, and it makes a nucleophilic attack on the carboxyl end of the growing polypeptide chain that is being attached with the 3' end of the tRNA. So what happens? A covalent linkage, a peptide bond between them is formed, a water molecule is released, and a CONH bond is formed like this. So this is how peptide bond synthesis occurs. The enzyme is basically peptidyl transferase, and that is what is that? That is present in the 23S rRNA of the large subunit of the ribosome. As you know that the ribosomes are basically RNPs, ribonucleoproteins. That means they contain RNAs and they are made up of proteins also. So very complex structure. We can have a look at the ribosome in some of the lecture. So this is how peptidyl transferase reaction occurs. Now let's move to the termination. In termination, now the growing polypeptide chain is attached with this tRNA, and the genetic code, that is the next code that has come, is not a genetic code, it is a stop codon rather. And for this, there will be three types of stop codons: UAA, UAG, and UGA. And for any of these three, there are no tRNAs in the system. So no tRNA will come and bind here. There is no tRNA for these three stop codons. And instead, there are release factors that come and bind with these termination sequences. And they hydrolyze the polypeptide at the 3' end of the tRNA. The carboxyl end of the polypeptide is hydrolyzed and the polypeptide is released like this. This is completed polypeptide or protein, you can say. It will go folding thereafter. So this is how termination occurs by binding of the release factor and hydrolysis process. Very simple. This was elongation, which we saw in the next stage in earlier slide. In earlier slide, that the elongation and this growing polypeptide, it has grown. This is empty tRNA that is being released. So in elongation phase, one after another, the tRNA brings a new amino acid, and that is being tagged with the growing polypeptide. The chain grows, and ultimately the stop codon arrives where there is no tRNA to bind there, and release factor binds, and it hydrolyzes the growing polypeptide. The already grown polypeptide, the full length polypeptide, is released and it undergoes for folding. And finally, the ribosome subunits are dismantled, and the mRNA is also released like this. And in fact, there are a number of ribosomes sitting one after another, and they are continuously synthesizing protein one after another, like a chain in a particular mRNA. But each of them, when they come to the termination site, they become dissociated like that, and they will go and again bind with the 5' end, and will start scanning to initiate the process. So this is the process of protein synthesis. Thank you very much for your patient listening. That's all about this.